Welcome to Planetterrian Daily, episode 129. It's July 23rd, 2026. Some interesting findings to cover today. A nature study maps the heart's independent neural network that modulates stress responses. Researchers describe a distributed set of neurons and ganglia embedded in cardiac tissue that operates separately from central autonomic control. The network adjusts local electrical activity and contractility during acute stress without requiring signals from the brain. Mapping shows these cells form a mesh that can sustain rhythmic output even when extrinsic nerves are interrupted. Follow-up work will test whether selective stimulation of this network alters arrhythmia risk in animal models. The study appears in Nature. The heart's intrinsic neural network operates with enough autonomy to sustain local rhythm control even when central inputs are blocked. This architecture adds a layer of resilience to cardiac function under stress and may help explain why some arrhythmias persist despite standard autonomic therapies. Mapping the network's molecular signatures could identify new targets for devices or drugs that act locally rather than systemically. Specific cell types within this cardiac mesh remain to be characterized in detail. Now, shifting to another example of distributed biological decision-making in the gut. Gut bacteria convert breast milk and formula nutrients into immune training signals according to a Nature study. The study tracks how microbial metabolism of specific oligosaccharides and lipids generates metabolites that shape T cell and B cell maturation in early life. Germ-free mouse models colonized with defined consortia demonstrated that the metabolites are required for normal mucosal immune development. Human cohort samples linked higher metabolite diversity to distinct antibody profiles at 6 months. The authors plan longitudinal follow-up to connect these early signals with later allergy incidents. Now here's something most people get wrong about how the immune system develops. Most people picture the immune system as a set of cells that simply learn to distinguish self from non-self after birth. In reality, right now, your intestinal bacteria are actively converting nutrients from milk or formula into metabolites that instruct developing immune cells on which responses to mount and which to tolerate. A single milliliter of breast milk can feed microbial communities that produce thousands of distinct signaling molecules, far outnumbering the immune cell types they help calibrate. The next time you consider how an infant's diet influences lifelong allergy risk, remember that the critical training happens through bacterial metabolism rather than direct nutrient absorption. Watch for upcoming human studies that track whether restoring specific metabolite profiles after antibiotic exposure reduces later autoimmune incidents. From microbial education of the immune system, another immune structure forms outside traditional lymphoid organs. Tertiary lymphoid structures receive expanded coverage in a new Nature paper. The report examines how TLS form at sites of chronic inflammation and support localized immune responses. Detailed imaging reveals organized zones of B cells, T cells, and antigen-presenting cells within non-lymphoid tissues. The structures appear in multiple disease contexts and correlate with altered tissue repair dynamics. Next analyses will track TLS persistence after resolution of the initiating stimulus. From organized immune architecture, a protein limits harmful protein spread inside the brain. A protective protein limits toxic tau propagation in mouse brain according to recent findings. Boosting levels of the protein reduced spread of pathological tau aggregates along neural circuits. The mechanism involves enhanced clearance or sequestration that interrupts prion-like transmission. Treated animals retained more normal synaptic markers compared with controls. The finding points to clearance pathways as potential intervention points in tauopathies. From neural protein management, a new genome editing method avoids the usual DNA break risks. A genome editing tool inserts large sequences into plants without double-strand breaks. Scientists at King Abdullah University of Science and Technology developed a method that directs integration of multi-kilobase DNA segments into rice and tobacco genomes. The approach bypasses traditional nuclease-induced breaks that often trigger unwanted rearrangements. Efficiency measurements show stable inheritance across generations in both species. The platform is now being tested for introduction of multi-gene metabolic pathways. From precise genetic insertion in plants, small changes in core cellular components can disrupt tissue architecture. Modest tubulin excess disrupts tissue architecture and cell viability in University of Geneva experiments. Even small overproduction of the microtubule subunit alters cytoskeletal organization in cultured cells and tissues. Excess tubulin shifts the balance of polymerization dynamics and impairs normal cell division and migration. Viability assays quantify reduced survival under the perturbed conditions. The results underscore dosage sensitivity for core cytoskeleton components. Phys.org covered experiments showing how magnetic fields shift electron patterns in a graphene-like material. Applied fields can tip the balance between striped and checkered charge density wave states. The competition arises from electron-electron interactions that produce collective electronic order. Precise field sweeps map the boundary between the two patterns and reveal hysteresis in the transition. The work supplies a controllable platform for studying how external parameters select among competing ground states. Climate-driven heat intensified recent European drought, according to an international analysis. The team attributes the severity of the drought primarily to elevated temperatures rather than rainfall deficits. Attribution modeling isolates the contribution of anthropogenic warming to soil moisture loss across the continent. The analysis covers multiple river basins and agricultural zones affected during the event. Future projections will incorporate updated temperature trajectories to refine risk estimates. National food waste diversion could cut emissions yet raise microplastic loads based on Vermont program data scaled to the United States. Life cycle modeling shows net climate benefits from avoided landfill methane alongside new contamination pathways through composting streams. Researchers quantified microplastic concentrations entering soils and waterways under different processing scenarios. Policy recommendations include upstream filtration steps at collection facilities. Endangered elkhorn coral moved to land-based hatchery ahead of warming in Florida. 30 fragments from offshore nurseries were relocated to a controlled facility to reduce exposure to predicted summer bleaching. The move targets a species already listed as threatened and vulnerable to cumulative heat stress. Hatchery conditions allow monitoring of symbiont retention and growth rates under stable temperatures. The team will compare survival metrics with remaining ocean cohorts through the season. Marine heat wave exposure includes extended warm periods before and after peak events, according to William and Mary researchers. They propose a cumulative exposure framework that accounts for the full thermal trajectory surrounding discrete heat waves. Analysis of historical records shows that preconditioning and recovery phases often contribute more total degree days than the core event itself. The metric alters estimates of ecosystem stress in regions with recurrent warming. Validation against long-term monitoring datasets is underway. Empty mollusk shells first colonized as habitats 470 million years ago. An international team dates the earliest recorded use of vacated shells by other marine organisms to the Ordovician. Fossil evidence from multiple sites shows small invertebrates occupying the interior spaces, creating a new ecological niche. The transition coincides with diversification of both mollusks and shell-dwelling taxa. Further sampling will test whether the behavior spread rapidly across ocean basins. Light-driven synthesis expands nucleoside analog libraries for antiviral screening at Simon Fraser University. Chemists report a photochemical route that generates 10 to 100-fold more variants than conventional methods within weeks. The analogs mimic DNA and RNA building blocks and have established activity against viruses and cancer. Library size increases allow broader structure activity mapping in cell-based assays. The team is now prioritizing compounds for testing against emerging pathogens. Water surface flow alone rotates floating microstructures without external power in work from CAT and the Chinese Academy of Sciences. Controlled vortices at an air-water interface can drive directional rotation of millimeter-scale objects. The system requires no magnets, electrodes, or chemical fuels. Rotation speed and direction are set by vortex geometry and surface tension parameters. Applications in microfluidics and soft robotics are under exploration. A phys.org article reports on a Chile-based longitudinal study of incarcerated women. Most women maintain stable low offending rates after release. Those with higher pre-incarceration involvement reduced offending when training or job placement was available. The analysis controls for prior criminal history and program participation. Employment emerged as a measurable factor separating desistance from persistence. Expanded program evaluation is planned across additional facilities. Before we go, keep an eye on survival metrics from the Elkhorn Coral Hatchery comparison with ocean cohorts. That's Planetterrian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. And if you'd rather watch than listen, find us on YouTube at NERA Network. Links in the show notes. This show comes to you from the NERA Network. If you want another sharp daily briefing, Unintended Consequences has you covered. True stories of good intentions that backfired. All our shows are free at neranetwork.com. New to the network? neranetwork.com/start-here picks the right daily briefing for you in about 2 minutes. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.